Assalamu alaikum students. I hope you will be fine at your homes. So in the previous lesson, we talked about the oxidative phosphorylation, which is the last and the fourth stage of aerobic respiration. Today, we have seen in the process of aerobic respiration that there are a number of hydrogen carrier molecules which are involved, and basically these hydrogen carrier molecules they accept the hydrogen whenever hydrogen is removed, and once they have accepted the hydrogen, they are going to become reduced. Then these reduced hydrogen carrier molecules, they are going to enter into the last stage of aerobic respiration, where we have seen what happens to these reduced hydrogen carrier molecules. Today we are going to discuss about the various types of hydrogen carrier molecules in the syllabus. As per your syllabus, there are three different types of hydrogen carrier molecules. It's NAD, it's NADP, and it's FAD. So basically, we have to see what are their roles very, very briefly over here, because we have seen the detail of NAD in the process of oxidative phosphorylation. Same goes with FAD as well, right? NADP we will be discussing later on. So basically, remember this thing that you need not to memorize the structural formula of these hydrogen carrier molecules. I mean, you need not to draw the structural formulas. You just should be knowing what are the various components of each hydrogen carrier molecules, like why they are named so. You can simply say, but as far as their structural formulas or their drawing is concerned, that need not to be memorized. So first of all, we will come to the NAD. Now, we have to see what does NAD stand for. NAD stands for Nicotinamide Adenine Dinucleotide. And to some extent, this name explains what are the various components of NAD. Now, basically, if we look at this, where NAD is used, basically NAD is used as a hydrogen carrier molecule in the various stages of aerobic respiration. Like NAD is involved in glycolysis, NAD is involved in link reaction, and it is there in Krebs cycle as well. So basically, in the first three stages of aerobic respiration, glycolysis, link reaction, and Krebs cycle, NAD is there. So if you see that, if we look at their name, nicotinamide adenine dinucleotide, first of all, we will come to this portion of the name, dinucleotide. It means NAD consists of two nucleotides. Di means two, nucleotide word comes from the nucleotides. And now we know that each nucleotide, basically, when we have studied nucleotide in DNA and RNA. When we talk about a nucleotide, we say each nucleotide consists of one sugar, one nitrogenous base, and a phosphate group. Okay, so same is the case over here as well. As we say, NAD consists of two nucleotides. Now we have to see what is present in each nucleotide. Basically, in both the nucleotides, what are the same components? In both the nucleotide, the sugar which is present, that remains ribose, right? Then if we talk about phosphate group, obviously phosphate group remains the same in both the nucleotides. So both the nucleotides, they have one phosphate group attached to each nucleotide, and in both the nucleotides, the sugar which is present, that is ribose sugar. So then we come to the next one. Now, what is the difference there between the two nucleotides? And actually that difference comes in the name, nicotinamide adenine dinucleotide. Now we come to the word adenine. Adenine is obviously one of the nitrogenous bases. So basically in one of the nucleotides of DNA, the nitrogenous base adenine is present. So it means in one nucleotide, there is a ribose sugar attached to a phosphate group and to the nitrogenous base adenine. While if we look at the second nucleotide, instead of having adenine, like it doesn't have adenine, in place of that, it contains a ring, which is nicotinamide ring. So then if we look at the second nucleotide, it means what is present in the second nucleotide? A phosphate group attached to the ribose sugar, and instead of adenine, it has a nicotinamide ring. And that's why you see that comes in the name of NAD as well. Nicotinamide adenine dinucleotide. Dinucleotide means it consists of two nucleotides, and obviously as both the nucleotides, they have the ribose sugar and phosphate groups attached. so no need to mention it in the name. What comes the difference between the two nucleotides? In one nucleotide, nitrogenous base adenine is present, while in the other nucleotide, nicotinamide ring is present. So that's why the name is there, nicotinamide adenine dinucleotide. Now you see, basically, um, uh, you see, whenever we say that NAD becomes reduced, actually, when NAD becomes reduced, it accepts one hydrogen ion and two electrons, okay, and then it becomes reduced. And actually, where is the place where this NAD is going to accept the hydrogen ion and two electrons? It's the place on nicotinamide ring, right? So after nicotinamide ring accepts the hydrogen ion and two electrons, the NAD becomes reduced. Now you will see the structure over here, NAD, and the equation for the reduction of NAD. You can see NAD plus, this is the oxidized form of NAD. Once it accepts, obviously, when hydrogen is removed, you see when hydrogen is removed, hydrogen, now what does hydrogen contains? It contains two hydrogen atoms. Each hydrogen atom has one hydrogen ion and one electron, right? So basically there are two electrons, two hydrogen and ions. And as we know that for the reduction of NAD, it has to accept one hydrogen ion and two electrons. So it accepts two electrons and one hydrogen ion at nicotinamide ring, and it becomes reduced. So what is left behind? That is one hydrogen ion. So actually, you see, this is the place. This is like you can see over here. This is one nucleotide. This is the second nucleotide, right? And if you see this nucleotide, it has a phosphate group. It has adenine nitrogenous base. While this is the second, and this is ribose sugar present over here. And this is the second nucleotide, which has a ribose sugar, a phosphate group, and a nicotinamide ring. And actually, this is the place at nicotinamide ring, which is going to accept the hydrogen ion and two electrons. So this is the structural formula of NAD. But as I told you earlier, you need not to memorize the structural formula. Okay, so this was all about nicotinamide adenine dinucleotide. Once again, I will explain it. It is acting as a hydrogen carrier molecule in the process of aerobic respiration, and it is involved in all three first stages. Like it's there in glycolysis, link reaction, and Krebs cycle. And then from all these three stages, reduced energy is going to enter all the reduced energy molecules. They're going to enter into the electron transport chain. Then next comes NADP. Now you can see over here, we can understand from the word NADP, how is it different from NAD? It's only the addition of this P over there. So actually when we see that for what NADP stands for, it's nicotinamide adenine dinucleotide phosphate. So it means it's not much different from NAD. Once again, consisting of two nucleotides, just like NAD. And in both the nucleotides, you see, obviously, ribose sugar is there. In both the nucleotides, phosphate group is attached to each ribose sugar. And in one nucleotide, nitrogenous base adenine is present, while in the other nucleotide, nicotinamide ring is present. So basically, you can see these structural details are all similar to NAD. Then how is it different from NAD? It is slightly different from NAD. And you can see over here that if we look at the structure of NAD back to this, you can see that actually over here at carbon number 1 of one of the ribose sugars, basically what is present, that instead of hydrogen, there is a phosphate group present over here. So this is the carbon number 1. So basically there is a phosphate group present on carbon 1 in one of the ribose sugars instead of hydrogen. Okay, so actually this is the place where a phosphate group is present instead of hydrogen. So that's why this is called as NADP, nicotinamide adenine dinucleotide phosphate. So you can see it is different from NAD just in having a phosphate group on carbon number 1 of one of the ribose sugars instead of hydrogen. That's it. Otherwise, it's exactly similar to, NAD. And as far as the function is concerned, but this is a major difference from NAD. NAD is a hydrogen carrier while NADP is used as a hydrogen carrier molecule in the process of photosynthesis, right? So I hope this is all clear. Then we come to the third hydrogen carrier molecule, which is FAD. FAD stands for flavin adenine dinucleotide. Once again, you can see over here, dinucleotide word indicates it again consists of two nucleotides, right? First of all, if we come to the function of FAD over here first before coming to the structure, because function is all similar to NAD, right? FAD is acting as a hydrogen carrier molecule in the process of aerobic respiration, but its involvement is only there in Krebs cycle. It's not involved in glycolysis, it's not involved in ring reaction, it's only there in the Krebs cycle where it becomes reduced, and when it's become reduced, then this reduced FAD molecules, there are two reduced FAD molecules in Krebs cycle which are going to enter into the oxidative phosphorylation. So once again, if we come to their structure, it consists of two nucleotides. One nucleotide, once again, it contains the ribose, the phosphate group, and adenine. While if we look at the, so obviously over here, up till here, structure is similar to NAD. But when we come to the second nucleotide actually it contains a very unusual structure which involves a linear molecule which is called as Ribitol instead of ribose, right? So this is how FAD is different from NAD. So this was all about the different hydrogen carrier molecules. I hope it's all clear to you. Then we next come to the synthesis of ATP. If you look at the book, when we started the process of respiration, remember we skipped synthesis of ATP and the role of active transport, and we moved straightforwardly to the process of respiration. Because I told you at that time, it's better we study the process of respiration in advance, and then we come to the detail of synthesis of ATP and obviously the role of ATP in active transport. So today we are going to study about the synthesis of ATP. Now you see, obviously for the synthesis of ATP, once again we say energy is also required, right? So basically that energy which is required for the process of synthesis, it can be available in two ways. Like there can be two types of energies which are used in the process of synthesis of ATP. One is the chemical potential energy, second is the electrical potential energy. Basically these two types of energies are released in the process of respiration, and then the synthesis of ATP takes place. But these are the two different types of energies there. It's chemical potential energy and electrical potential energy. So now we have to see where chemical potential energy is released and where electrical potential energy is released. So first of all, coming to the chemical potential energy. If you remember, when we studied the process of glycolysis and you see Krebs cycle, then we saw there that in case of glycolysis, 4 ATP molecules were made, right? From ADP and inorganic phosphate. In the same way, in the Krebs cycle, 2 molecules of ATP, they were made once again, obviously from ADP and inorganic phosphate. So basically, the chemical potential energy is released during glycolysis and Krebs cycle. You don't see, obviously in link reaction, there is no production of ATP molecules there. So no chemical potential energy there, no electrical potential energy there. When we talk about glycolysis and Krebs cycle, no electrical potential energy released over here. It's only chemical potential energy which is released during the process of glycolysis and Krebs cycle. And basically, you see, this chemical potential energy is released when there is a reorganization of chemical bonds during the process of glycolysis and Krebs cycle. So we have seen on the way in the process of glycolysis, in the process of Krebs cycle, that various compounds, they are converted into some other compounds. Some of them they are the isomers, and obviously there are the structural changes. There is a rearrangement of items, so there is a reorganization of chemical bonds. During this reorganization of chemical bonds during the process of glycolysis and Krebs cycle, the energy which is released, obviously that is chemical potential energy, because that is going to be released reorganization of chemical bonds, right? And then that chemical potential energy is used for the synthesis of ATP molecules, which were 4 in case of glycolysis and 2 in case of Krebs cycle. So we can see a small number of ATP molecules are made. Like if you remember the process of glycolysis, those 4 ATP molecules are made there, but as 2 are utilized, so we say net gain is 2 ATP molecules. So we consider it in terms of 2 ATP molecules in glycolysis and 2 ATP molecules in Krebs cycle. We can see that a small number of ATP molecules are made using this chemical potential energy. Okay. Now, what is substrate level phosphorylation? Now, you can understand that when ADP and inorganic phosphate combine, what is formed ATP? So actually, what is happening to ADP? It is phosphorylation of ADP which is taking place, because the phosphate group is going to be attached with ADP and it is converted to ATP. So this is phosphorylation. Wherever there is the direct production of ATP using the chemical potential energy, that type of phosphorylation is called as substrate level phosphorylation. This word is not, this terminology is not used in the book anywhere, but it often comes in the Cambridge question paper. Okay, they often ask you that, sometimes they ask you where substrate level phosphorylation takes place, and sometimes they, let's suppose it's the process of glycolysis and where ATP is made, they are going to ask you to label the stage, right? So this is going to be substrate level phosphorylation. So remember that phosphorylation of what? Phosphorylation of ADP, because a phosphate group is going to be attached with ADP and that is going to result in the formation of ATP. And wherever there is a phosphorylation of ADP means the formation of ATP using chemical potential energy, that type of phosphorylation is called as substrate level phosphorylation. Okay, so I hope it's clear now. So this was all about chemical potential energy. We have seen that very small number of ATP molecules are made using this chemical potential energy, which is released during glycolysis and Krebs cycle. Then we come to the electrical potential energy. First of all, where electrical potential energy is released, obviously it's released in the last stage of respiration. This is oxidative phosphorylation or electron transport chain. Now, if you remember, we saw on that day that all the reduced NAD and FAD, when they enter into the process of oxidative phosphorylation and then they undergo the detailed process, and then the process of chemiosmosis takes place, then you see there is a synthesis of ATP molecules. And if you remember, we saw on that balance sheet as well that how many molecules are of ATP they are made in oxidative phosphorylation. They were total 28 in number. So we can imagine, as compared to 4 number of ATP molecules which were made in glycolysis and Krebs cycle using chemical potential energy, you can compare the number of ATP molecules made in oxidative phosphorylation, where for the synthesis of ATP, what type of energy is involved? That is the electrical potential energy. So we can see the large number of ATP molecules they are made using this electrical potential energy. This electrical potential energy is released during the process of oxidative phosphorylation. And remember this thing, that this process of oxidative phosphorylation, it occurs in both mitochondria and chloroplast. In syllabus, we are just concerned with its detail in mitochondria, while when we will be talking about the process of photosynthesis, then we can talk about chloroplast in this reference a bit detail. Okay. So we have already studied the process of oxidative phosphorylation, that basically how this electrical potential energy is released. This is because when the electrons are transferred, by the electron carriers in on the and this process takes place on the inner mitochondrial membrane. And you see, when electrons are transferred from one electron carrier to the next one, down the energy level, energy is obviously released, and that is the electrical potential energy. And we also know that some of the energy is utilized to move the protons from the matrix into the space present between the inner and outer mitochondrial space. And once there is a buildup of high concentration of proteins in that intermembranous space. Then the protons, they start diffusing back into the matrix by the process of facilitated diffusion through the protein channels, and they are the transmembrane proteins, right? And part of that transmembrane protein is acting as an enzyme which is called as ATP synthase. So basically, I have written the few outlines over here, that there is the transfer of electrons by the electron carriers, energy is released, and then there is a concentration gradient for protons. Obviously, I haven't written the details of this. Then you see, when protons are moving back into the matrix by the process of facilitated diffusion, then ATP synthase, as it is a part of that protein through which protons are moving. So, and this ATP synthase is the enzyme which is used, used to catalyze the synthesis of ATP from ADP and inorganic phosphate. Generally, it is believed that when there is a transfer of 3 protons, that results in the production of 1 ATP molecules. But once again, it depends on whether ATP and inorganic phosphate, they are present inside the organelle. Okay, then the 3 protons are, when they are transferred from the intermembranous space into the matrix, then 1 ATP molecule is made. When we were talking about the oxidative phosphorylation, we know that this process is called as chemiosmosis, and this process was actually proposed by a scientist whose name was Peter Michel in 1961. So once we can see in this diagram as well that they have just shown the part of that protein which is acting as a transmembrane protein. You can see over here, this is a phospholipid bilayer, and this is the intermembranous space present over here. And once there is a high concentration of hydrogen ions, then hydrogen ions, they are going to diffuse back into the matrix down the concentration gradient by the process of facilitated diffusion through this transmembrane proteins. And you can see over here, for every 3 hydrogen ions or for every 3 protons moving into the matrix, there is a synthesis of 1 ATP molecule. And a part of this protein is acting as an enzyme ATP synthase, and this ATP synthase, as the name indicates, it is involved in the, it is going to catalyze, it is going to speed up this chemical reaction. It is acting as an enzyme in the process of synthesis of ATP. That's why it's named so as well. Now we will come to this. Now there is, you can see even in the book, there the structure of ATP synthase is given over there. If we look at the structure of ATP synthase, unfortunately, I could not upload the diagram of ATP synthase. I will be doing it in the next lesson. Then we can have a review of that. But you can, for the time being, you can look at the books as well. ATP synthase is actually having 3 binding sites, right? And it has, along with 3 binding sites, it has a rotating portion present in the middle, which is going to rotate. And actually, the 3 binding sites, they're attached to the rotating portion. So when the portion is, so when that portion is going to rotate, the 3 binding sites are going to change their position. So basically, there are 3 phases involved in the process of ATP synthesis involving ATP synthase. First of all, binding of ADP and inorganic phosphate takes place. Second step is that obviously it keeps on holding this ATP tightly, and then in the next third phase, it releases the ATP into the the matrix. It is just like this. We can take the example of that rotating gates, which are present on certain shopping malls. If you have seen, when you're going to enter into that, then you're going to move down, and it's going to move. It's going to rotate, and you get free on the other side. It's just like that. So we can have a detail of this in the next lesson, along with the role of ATP in active transport. I hope the things are clear. If anything is not clear, please, you can call me anytime. You can call me on WhatsApp. You can put your question on in that group on WhatsApp, right? If you want to talk to me personally, you can call me then. I'm going to have a worksheet attached with this lesson as well. I will be checking your all previous worksheets, whatever you people have submitted to me by this weekend, and kindly send me this worksheet as well. So see you people on next Tuesday.